I'm Daniel. I'm a final year PhD student in the Keras Lab, and I'm here to give you all a yearly reminder that Acholeplasma is still a thing that we do. Acholeplasma research is still going on. So this is not really a talk about the minimal cell or any kind of minimal cell, but this is more a talk on our efforts to extend the technology that made creation of the minimal cell possible, that being whole genome cloning and transplantation, to this new species, Acholeplasma laidlawii. And I'd like to start with a justification for why we want to use Acholeplasma when we can already do genome transplantation in mycoplasmas. Firstly, Acholeplasma is unique in the sense that it is a molecule, but it uses a standard genetic code. And to date, there are no transplantable species that have this trait. Additionally, we actually in Canada cannot work with some of these strains used for genome transplantation because of their high biosafety level. Whereas Acholeplasma we can use in Canada, it's a biosafety level 2 species, and I believe in other parts of the world it's BSL1. Also unique for Acholeplasma is that it does not require the growth of any kinds of serums, although as we heard in an earlier talk, of serum-free media for mycoplasmas now exists, so maybe this is a little bit less relevant. Although throughout the talks, I was also reminded that CRML is also usually included in SP4, and we do not include this in our SP4 for Acholeplasma, and the cells seem to grow fine. So perhaps I'm shifting the goalpost here a little bit. But importantly, Acholeplasma laidlawii is still a molecule, so it has these properties of having a small genome and not having a cell wall that we're hoping will make the adaptation of whole genome replacement from mycoplasma to Acholeplasma less difficult if we can still take advantage of these traits. This research is mainly confined to two species of Acholeplasma laidlawii. So we have here strain PG8A, which is probably the best studied Acholeplasma strain. This is the strain that Bogomil showed whole genome cloning for in yeast. And we have both suffered quite a bit before realizing that this strain is extremely resistant to transformation. And for that reason, we have moved over to using this other strain, 8195, for a lot of our genetic manipulation work. This strain can be transformed, and also it is suggested to be both restriction and recombination deficient, and that makes it a really good recipient for genome transplantation. So ultimately, we are hoping to enable the transplantation of the PG8A genome into 8195. And I want to emphasize that when I was first starting out in the lab, we had all these big goals for working with Acholeplasma. But back in 2020, when I started, we were basically starting from scratch. I didn't even have a recipe or a protocol for how to grow the strains. So there was a lot of foundational work that had to be done before I could start on more of this complex stuff. And I'd like to briefly talk about some of the groundwork that I've laid for this project. So my first chapter was genetic toolbox development for laidlawii. Towards this goal, I have created a few genetic tools for this strain, mostly for strain 8195, including transformation of different plasmids that have different antibiotic markers and replicons. These plasmids can be transformed through different transformation methods. We mostly use electroporation, but I have also shown bacterial conjugation and PEG-mediated transformation. We can also use the TN5 transposase to integrate antibiotic markers randomly into the genome. And I have also shown one instance of transformation of this marker to strain PG8A. So this could be useful as a transplantation donor later down the line. And finally, I have evolved a strain of laidlawii 8195 that we call DNE. And this strain shows a increased transformation efficiency via electroporation. We see around a 100 to 200 times improvement in transformation for both of our plasmids. And with some of these basic genetic tools established, I was able to move on to some more complex work. So my second research chapter was whole genome cloning. We felt it was important to replicate the work that Bogomil had done previously in cloning the PG8A genome and try cloning our evolved 8195 strain, DNE, in yeast. And to keep things as a summary, there were a few barriers that I had to overcome. So firstly, I had to complement this strain with Rec-A in order to restore the ability of it to do homologous recombination. And then I was able to integrate a yeast vector directly into a gene that we know is toxic to yeast. Once this strain was generated, I was able to move the genome to yeast through cell fusion, which is just mixing the cells with yeast spheroplasts. And here is just some evidence showing that cloning of the whole genome appears to be successful. Here on the left, I have a map of the 1.46 megabase genome, and we have different multiplex primers that bind to various regions of the genome. So when we do cell fusion using Acholeplasma that has the yeast vector inserted directly into the toxic gene, we have yeast that screen positive for the whole genome. But when we do cell fusion using Acholeplasma that has the yeast elements inserted, but the elements are inserted elsewhere to keep the toxic gene intact, we do not see any instance of whole genome cloning. And after several years of work, this finally brings us to what we wanted to do initially, which is establishing genome transplantation in Acholeplasma. Towards this goal, I had the privilege of spending 1 month at the JCVI in San Diego to learn how to do transplantation in mycoplasmas. Of course, Bogomil knows how to do transplantation, but there was this issue with bringing the mycoplasmas over because of the biosafety consideration. So it was actually easier for me to go to the mycoplasmas in this case. And here I have some results showing genome transplantation experiments I've done moving the Mycoides Syn 1.0 genome into Mycoplasma capricolum. And I can now confidently say that I've picked up the technical knowledge on how to do transplantation, at least for mycoplasmas. And I would like to spend the next few minutes discussing some potential considerations we have for obtaining our goal of genome transplantation, specifically for Acholeplasma. Firstly is this consideration of cell fusion. In the very first transplantation paper, it is noted that the transplantation protocol appears to promote mycoplasma fusion. And whether this is a coincidental thing where the protocol also just allows for cells to fuse, or whether this is actually necessary for transplantation to happen, we think that if we can find different parameters that promote cell fusion between Acholeplasma, then we're probably on the right track for developing a transplantation protocol. And our method of determining whether cell fusion has occurred is similar to what is noted in that first paper. We have a tetracycline-resistant strain of Acholeplasma bearing a marker in the genome, and we have a neomycin-resistant strain carrying PNZ18, and we're hoping that if we mix these cells together in the presence of PEG, then we will be able to pick up individual strains that are both tetracycline and neomycin-resistant. And it seems like we already have some promising preliminary results towards that. This is an experiment that Bogomil did, actually. He was so excited about how close we are to transplantation, he got into the lab and started doing some fusion experiments. So this is a combination of those two strains I showed on the last slide, where Bogomil mixed them with different concentrations of PEG and then plated them out in serial dilutions with double antibiotic selection. And then I picked out some culture from one of these wells, moved them onto solid media to pick up individual colonies, and then I screened them using PCR. And we're happy to see that in the beginning, we only had strains that tested positive for one of the two markers, but following this fusion protocol, we end up with strains that test positive for both. And we're also happy to see that this happened both when we included serum in the media and when we didn't have any serum present. Our second consideration for genome transplantation in Acholeplasma is extracellular nucleases. We have observed very potent extracellular nucleases in both strains of Acholeplasma, PG8A and 8195. You can see that when we incubate live cells with PUC19 DNA, even after 30 minutes of incubation, most of the plasmids are destroyed. And of course, this is a huge consideration for when we're incubating cells with whole genomes for upwards of an hour. Luckily, it seems like these nucleases are a little bit less active in 8195, so we're already on the right track. But we still have to consider ways that we can inactivate or at least reduce the activity of these nucleases. And we have a fourth year student joining us, Isabel, who is going to be testing some different compounds. And hopefully we will find some that don't kill the cells and don't stop the PEG transformation from happening, but reduce the activity of these nucleases. Some things we are considering are trypsin, which John had mentioned to us previously, EDTA. As you can see here, when we add 10 millimolar EDTA, the digestion of the DNA stops. And also, we are considering things like carrier DNA or yeast tRNA. And with that, I would like to give some acknowledgements to Bogomil and the rest of my advisory committee, all past and present members of my lab, the people who have helped me author these papers, and also thank you to the GLASS team at JCVI for being welcoming of me as I was there back in March, and especially thank you to Zumra and Sage for teaching me how to do transplantation. And finally, thank you to the members of our Acholeplasma working group. We meet every so often to discuss research with Acholeplasma, and without the exchange of protocols or resources, I would have never gotten anything to work with Acholeplasma. And finally, I will say that if you are interested in doing any kind of work with Acholeplasma, you don't have to wait for us to develop transplantation. We already have quite a few strains, plasmids, promoters, markers that are available for use. So if that's something that interests you, feel free to reach out to me or Bogomil. And also, we are looking for some PhD students to hopefully continue this work of working towards developing synthetic Acholeplasma strains. So contact Bogomil if that's something that interests you. And thanks.
Very nice, Daniel. So if you treat cells with EDTA, does that eliminate your ability to transform those cells?
I think yes, depending on how much EDTA we add. So I had previously shown that if we add a little bit of EDTA prior to electroporation, the electroporation still works, and it seems to, in some cases, work a little bit better. But at the same time, I've definitely added too much EDTA to the point where I'm starting to lyse the cells. And also, according to some previous literature, EDTA, or the removal of ions, seems to decrease the level of cell fusion. So I think there might be a sweet spot here where we can inactivate or reduce nuclease activity while still keeping the cells and the protocol intact.
I mean, it's still, it seems like it would be more important to understand how it affects a chemical transformation, because the calcium that you add has the purpose of masking the charges on the nucleic acid and the cell. And so if you add the EDTA, that phenomenon may go away. Other questions for Daniel?
Beautiful work, Daniel. James Pelletier here. I'm curious, when you evolved the strain DNE, how did you select for higher transformation efficiency?
So what I did was I had seen in some literature that naturally when people transformed and cured Acholeplasma strains, they would sometimes end up with strains that had more transformable phenotypes. So what I did is I started with a single culture, like originating from one strain of Acholeplasma, and I would continuously transform it and then cure it of the plasmid. So I did that transformation curing cycle 4 times total. And then following that, I took the final culture of the cured cells. I plated out for individual colonies, and then I just started testing them for transformation alongside the wild type to see if we can find any that had greater transformation efficiency. And it seems like quite a few of the final strains did have an improved transformation phenotype.
Very cool. Thank you.
Thanks.